My name is Nick Geraghty and I am a post-doctoral fellow working at Molecular Horizons under the supervision of Mark Wilson. My project uses a cell-based model to look for drugs that reduce the amount of proteins in a cell that cause a number of diseases. So normally in a cell you have the proteins that are made, they fold correctly, they go and do their job, and when they can't do their job they're basically broken down and taken away again. Unfortunately, sometimes when the proteins are made, they don't quite fold correctly, so this leads them to stick together and form big clumps, what we know as aggregates. And these protein aggregates can cause lots of problems in a cell and stop the cell from working properly, and can eventually lead to cell death. So this is implicated in some diseases, such as Alzheimer's disease, where these protein clumps or aggregates stop the messages from going around the brain, or in diseases such as motor neuron disease, notably suffered by Dr. Stephen Hawking, where the body starts to lose its ability to control the muscles and because the proteins stop the electrical signals being sent down. So what I do is, using these cells, I introduce the protein into these cells, force it to misfold and form all these clumps, and then I put these cells across a plate of novel drugs that have never been tested before, and hopefully I incubate these for 48 hours to look for any drugs that potentially reduce the amount of these protein aggregates in the hope to eventually find a cure for these diseases. So I do this using flow cytometry, which is a machine here that measures the amount of protein stuck together because we've tagged it with a fluorescent protein, and I can look at that on the screen by looking at the amount of reduction of inclusions, which is the protein sticking together.